Matrix and welcome to Matrix. My name is Looney and today we'll be assisting you with your final exam prep for life sciences. All you need to do to get your questions and comments through to us is hit us up on our social media platforms, search for Matrix as well as our WhatsApp line. All of the details are on the screen. Guys, please don't forget to take part in our assessment just by downloading the 2Enable app for free on your app store. We also have a cool competition going on, so stay tuned to get all of those details. With me are Nicolene, our sign language interpreter, as well as Eugen, our awesome teacher. Thank you so much, guys, and over to you.
Thanks, Looney. Welcome, Matrix. So, folks, we into that preparation for that final paper 2. So we're going to look at Life Sciences paper 2 today. I've put together a presentation which has questions from a few different exams that have been written already. And I'm going to kind of engage with you on those questions, but try and develop some content background to that. Remember, guys, the questions that have been posted on the app, please download them. Try and solve those questions, send in your answers. At the end of this segment, we're going to look at your answers and we're going to try and unpack some of the questions that you've kind of struggled with and possibly look at the misconceptions that you have between answers in a multiple choice, which often happens when you read through them and you're rushing right at the start of the exam and you've kind of stressed about those questions that are still to come. So let's get into those questions that I've posted on the app. Let's look at the first one, and I challenge you to give me your answers and I'll give you feedback on that at the end of this session. So the first question refers to the following. The diagram shows homologous chromosomes. So guys, the concept of homologous chromosomes, remember, similar shape and having genes at the similar position. With a pair of number 3 from each of 4 different fruit flies, the alleles for the antennae shape are normal or abnormal, and the body pattern with stripes or without stripes are indicated on the chromosomes. So that's kind of an information that's given to you about what we call fruit flies, and that have alleles that are either normal or abnormal for the antenna shape, and then that have either stripes or no stripes for their body pattern. Okay, let's look at this diagram. So here you've got the homologous pair for fly number 1, a pair for fly number 2, 3, and 4, and a key that's given here that indicates the shape or the color code for the normal antennae and the abnormal, as well as the body with stripes and the body without stripes. So again, referring to the question, let's go and identify the following. So which fly is homozygous for antennae? Again, the word homozygous, you should come to your understanding now that they have both the same alleles, and heterozygous for body pattern. So you've got to identify which fly is homozygous for antennae and heterozygous for body pattern. And so guys, that's the challenge question. It will require that you go back and you look at the diagram and try and understand and link together what homologous pairs are, what homologous and heterozygous are in terms of when alleles are together in a genotype, and use that to identify this. And I wish you well with this question. It's quite a challenging question. I'm hoping that many of you get it right, and those of you that don't, we're hoping to be able to feed back to you so that you're able to understand why you've got it wrong and probably improve your understanding around how chromosomes are inherited and the variations we experience when alleles recombine together. Right. So Matrix, a question that was taken from the September 2020 question 2, which I'm going to go through now. So we have a diagram here that I've illustrated. Let's study the diagram and answer the questions that follow. And if you look at this diagram, you've seen many of these, and these questions often seem to come through in an exam, especially in paper 2. There hasn't been a question that has been left out with transcription or translation that we must. So learn the section well. So it's a diagram that shows you the nucleus, which is clearly marked and labeled for you here. You've got a structure labeled number 1, and I'm not going to call that out now. And then you've got a structure here that is shown to be leaving the nucleus, and that then moves into the area outside the nucleus, which we know is the cytoplasm. You then see structures here that are putting together substances together, and these structures are labeled number 2, 4, 3, and 5. And while we're at it, let's look at these structures. So we know that in this diagram, there's a process that's being illustrated that is responsible for the production of this structure here. And that structure that we're looking at is called mRNA. Okay? And the process that is occurring here is the first step of protein synthesis, which is known as, if you remember correct, yes, transcription. And so transcription is the process that involves the production of mRNA, and that occurs inside the nucleus. The next question, the next part or phase of this would be what happens outside in the cytoplasm, and that's the production of these amino acids held together, and we refer to that as the second stage of it, called translation. And translation is essentially changing the mRNA into a polypeptide chain which has many amino acids held together in a specific order, and that is essentially what number 3 is pointing to. We know that we've got structures that play a very important role in transporting those amino acids to the site of protein synthesis, which are called tRNA, and this little structure here, which actually reads the mRNA, is called the ribosome. And ribosome is the third type of RNA, which is also sometimes referred to as the rRNA. And so that is essentially the structures that we've identified. We also know that they've asked us to identify, probably ask us to identify structure number 4, which we know is the mRNA. And they're pointing specifically to, well, this structure here, which is on the tRNA. And I know that on the tRNA, we have the presence of anticodons, which are the triplet base pairs on a tRNA molecule. So that's the anticodon. Okay, so let's look at the questions that are based on this diagram. So we've gone through lots of the diagram. We've kind of interpreted the question, the images. We've labeled what we needed to. Let's now answer these questions. Name the process represented by A and B combined. So again, if you look at the mark allocation, that's a 1-mark question. So we've got to name the processes indicated by A and B. And so A is kind of that process there, B. And collectively, we know that this is pointing to specifically, or as a whole, the process of protein synthesis. And essentially, protein synthesis is the synthesis or the manufacture of proteins, where we take the amino acids, as indicated here, and we put them into a specific sequence based on the information that comes from the DNA. And so this, collectively, A and B are known as protein synthesis. An interesting question. Where in the cell does process B occur? So if we go back to that diagram, if we look at process B, this is a large cell, guys, and if I were to kind of complete the diagram, we actually inside the cytoplasm, we inside the cell, and here we're seeing the nucleus, and so outside the nucleus, we're in this area, and this area is referred to as the cytoplasm. So it's the contents outside the nucleus, which is the cytoplasm. Cool. More questions on this. Identify the molecule, the following molecules. Okay, polymer 3, and I'm sure this points to molecules. Let's look at the diagram. So we've gone through very quickly. We're looking at molecules 3, 5, and 4. So if we were to look at them, we've clearly identified these as a polypeptide chain, and that's essentially pointing to this structure number 3, which is made up of many amino acids which are held together in a chain called a polypeptide chain, which will eventually be joined with other polypeptides to form a protein. So polypeptides held together form proteins. We know that number 5 is the monomers of proteins, which are called amino acids. So these amino acids basically are what are held together by peptide bonds that hold them together here. So here we have our peptide bonds that join them together. That's an important bond that holds the amino acids together. So you have your peptide bonds. And this structure here would be the mRNA, which I described earlier on, and we've mentioned the anticodons on the tRNA. So that covers all parts that require to be labeled on this. Okay, so we're going to move on to the next, so we've kind of discussed that. Let's move on to the next question. So here we've got information about the anticodons for tRNA and the corresponding amino acids that are coded for by these anticodons. So what's important to note is that these are anticodons, and anticodons are found in tRNA, and this table represents the corresponding amino acids for specific anticodons. So using this table, let's move on to the next question. What is the DNA base triplet of glycine? And I'm going to go back to this table and let's look at what glycine is. So glycine is coded for by CCA, and that is a tRNA, guys. So note that. So glycine has a CCA. I'm just going to skip back to that. So am I right? CCA, and we said that it's tRNA. Okay. So if it's CCA and if that is tRNA, okay, the question wants us to get the DNA base. So if we go back to what this image looks like, so that is the tRNA, and on it is CCA. Okay. You know that CCA corresponds to a specific, what we refer to as codons. So that codon would be, if we go and apply our rules of complementary base pair, that would be a G, and A base pairs with uracil. There's no thymine in mRNA. So that's the mRNA codon. Have we answered the question? The question points to DNA base. No. So we've got to take this mRNA codon and convert that into a DNA base triplet. Using this, remember that we've got a sequence here that codes for, let's go, G, U. We've got to establish exactly what the DNA sequence is that gives us that. So again, the concept of complementary, so that's going to be C, and we need to have on this an adenine. So those DNA bases would code for this specific mRNA codon, which in turn gives you the anticodon, which is CCA. So the answer for this question would be CCA. Yes, it is the same as the tRNA anticodon. Not a coincidence, but again, if we use our rationale of complementary base pairing, the first step was to convert the tRNA to mRNA, and then we used the mRNA in our second process to work out what the DNA sequence was. So again, this question requires you to use two processes. So it's a 2-mark question. It requires us to use two processes to be able to answer that question. Okay, let's move on to, so we've named the amino acid found at label 5. So I'm going to jump back to that diagram. So we've got to identify the amino acid found at label 5. So guys, let's go to that diagram first. And if we go to label 5, and if we look at this amino acid here, we've got to look at label 5 is a tRNA, and we need to identify that amino acid. When we look at this tRNA, it is complementary to a specific, in this case, codon, and that codon on the mRNA is AAA. Okay, so using this information, let's identify which amino acid is coded for by that tRNA. So the codon is AAA. Let's move back to our coding table. So we're using AAA as our codon. So I'm going to very quickly scribble this here. So remember that it's AAA, and AAA is what we know as mRNA. Okay? In order for us to determine the amino acid, this table gives us an anticodon table. So it's a tRNA anticodon. So we've got to convert the mRNA into its anticodon, and so we've got to use the complementary base pair rule. So that's going to be, again, help me guys, so that's going to be A would be with U, that's going to be A again U, and that will be U. So the anticodon for AAA is UUU, which points specifically to this, and hence our answer would be lysine. And notice that I had to do a couple of things. I had to first figure out what the mRNA was. I had to take that mRNA, put it back into the tRNA anticodon table, and figure out then what the complementary anticodon was. Once I found that, I was able to link that specifically to, in this case, the amino acid was lysine. So guys, we've kind of spent time looking at protein synthesis in this session. We've looked at being able to identify structures within the nucleus that are important in transcription. We've also looked at the concept of translation, structures known as tRNA, mRNA, ribosomes. We've looked at a codon, we've looked at anticodons, and we've used the codon table, in this case, the tRNA codon table. It's important that you recognize and understand what table has been provided. In some assessments, they give you an mRNA codon table. In others, like we've done today, it's a tRNA codon table. So that's important to identify. Before you start converting your codons, you need to identify what has been given. So look out for that. So guys, you've done well. I'm going to hand you over to Looney for a little break, and then I'll see you after that. Looney, over to you.
Thanks, Hugo. Guys, let's take a very quick break and we'll see you shortly after this. Welcome back from the break, Matrix. If you've just joined us, we are still doing your final exam prep to help with the upcoming exams. If you're someone who's constantly running out of data, then this next competition is just for you. Matrix is bringing you the hashtag WazzoWinner competition, where 2 lucky matriculants stand a chance to win 2 gigs of data. All you need to do to enter is head on over to our Facebook page and all of the details will be there. Thank you so much, Hugo, and over to you.
Thanks, Looney. Talking about data, guys, sure, I don't think data can ever be enough. So start, enter the competition. So let's get back to this question. A very interesting question. In fact, I would invite you to go and in fact, Google this question when I'm done and see how interesting this concept is. So we're going to get into genetics and we're going to look at the inheritance of characteristics. But here's a very new concept that you probably haven't heard of before, but I've fortunately come across an example of this before. And so I thought it would be an interesting question to share with you guys and to see some of the different disorders that are present around the world and that affect us. So also look at other examples locally. And I was chatting to the team during the break, and we spoke about a group of individuals in a tribe in Zimbabwe that have 3-toed people. So they're called the Vadumba tribe. So that's also a genetic disorder. So there are many genetic disorders that are relevant to South Africa and African context that could come in the exam. So let's look at this example that has been mentioned. Blue people is a genetic disorder caused by when a person does not produce an enzyme that converts methemoglobin, which is blue, into hemoglobin, which is what gives the blood a red color, or the hemoglobin, the red distinct color. When this happens, this results in a high level of methemoglobin in the blood. The result of this is that it causes the skin to turn blue, the lips purple, and the blood chocolate brown. So guys, here's a genetic disorder which has been inherited in a small isolated family that lives out in America, and this has been inherited over a few generations in that family. And essentially it was a mutation that took place that resulted in an enzyme that is responsible for converting methemoglobin into hemoglobin not being able to carry out its function. And this has resulted in significant phenotypic, so things that you can clearly see in them, changes. And in this case, you can see from that their skin appears blue, their blood appears chocolate brown. So let's look at the pattern of inheritance in this family and answer the questions based on that. So the diagram below shows members in the family who have blue skin. The genotypes of some of the members of the family are known and indicated on the diagram. So let's get into that diagram. So here we're seeing a diagram that shows the inheritance of this characteristic, and it tells us here right at the top that Billy Bob and Levi are cousins. So here you've got Billy Bob here, and you've got Levi that are cousins that have obviously married different individuals. So Billy Bob married Mary, and Levi married Sarah, and they've had kids. And over the generations, so that was the first generation, the second, and they've had kids separately. and their kids in turn have had kids in the 4th generation, which were Luna and John. So guys, here we're clearly seeing a family tree showing you the inheritance of characteristics from one generation to the next, and we know that this image or this illustration is called a pedigree chart because it shows you the inheritance of a characteristic from one generation to the next, and it's used often to study the inheritance of certain characteristics in a family over generations. Okay, so the first question, name the type of diagram shown, and as I mentioned, this type of diagram is called a pedigree chart. We often have other illustrations called karyotypes or cladograms, and those are different diagrams that illustrate relatedness or the process of evolution. A karyotype is used to look at the specific arrangements of chromosomes and to identify chromosomal abnormalities. A pedigree chart, as the one we've got in the diagram behind me, is an illustration that shows how a characteristic is inherited from parents to offspring and to the next generation. And so this particular diagram is called the pedigree chart. Okay, so as I showed you, it shows you how a characteristic is inherited from one generation to the next. Question 3.2. Give the following: the phenotype of Luna and the genotype of Hannah. Now guys, the importance of the word phenotype, remember, I often have to remind myself that phenotype refers to the physical appearance, so we're looking at the physical appearance of Luna, as well as the genetic, the genotype makeup of Hannah. So let's get back to the diagram. So we're going to look at Luna. Where's Luna? Here. So we need to look at Luna as well as Hannah. Am I right? Let me just go back one slide. So Hannah and Luna. So here's Hannah here, here's Luna here, and we've got to identify their phenotypes and genotypes. And while we're at it, let's look at both their phenotypes as well as their genotypes. Looking at this illustration, some of their genotypes have been given. In this case, Hilda has got a homozygous dominant allele combination, and Ben, in this case, has got a heterozygous allele combination. Knowing that the individuals that are affected will be shaded, we can come to the conclusion that Luna's genotype would be a recessive homozygous combination. So that would be her genotype, and her phenotype would be that she would be affected, meaning that she would be affected by this condition and her skin color will be blue. So we call this a homozygous recessive individual and an individual that shows a phenotype, in this case skin color being blue. Let's look at Hannah. And Hannah's quite interesting because she has an unshaded circle, so she's female, and we know that it should either be a dominant allele or with two dominant alleles, or a combination of a dominant and a recessive allele. So Hannah could be either one of these two. The question is, are we sure? Let's try and determine that. In order for us to determine what Hannah has as alleles, we've got to look at her parents. Billy Bob was affected, so he's got two recessive alleles, and Mary seems to be unaffected, so she certainly has a dominant allele. So the question that I'm going to pose out to you guys is, in order for Hannah to be unaffected, in this case unshaded, what would she have inherited from both the parents to have this? So the answer is quite obvious. So the only way she can have the dominant allele is if she's inherited a dominant allele from Mary. And so Mary has contributed to Hannah having the dominant allele. The next question is, well, then what would she have inherited from Billy Bob? When we look at Billy Bob is homozygous recessive, meaning that the only thing that he can possibly contribute to the offspring or his offsprings are a recessive allele. So we can clearly see that Billy Bob has passed down the recessive allele to Hannah, and hence Hannah can only be a heterozygous individual and not a homozygous dominant individual. Clearly because this dominant allele comes from Mary, the mother, and this recessive allele comes from Billy Bob, the dad. And so we've looked at the genotypes and phenotypes. And the clue to using a pedigree chart is to look at those individuals that are homozygous recessive. Essentially what I mean is those individuals that are affected by the condition, because you can work either backwards to their parents, or you can work towards the next generation and figure out what they pass on. So an individual like Billy Bob has to pass on the recessive allele to all his offsprings. So without a doubt, all his offsprings would have inherited a recessive allele. Likewise, if we look at Luna being affected means that the only way she could have been affected is if she gets a recessive allele from both the parents. So that is the only way Luna can be affected. And so these homozygous recessive individuals that have those alleles are important clues in a pedigree chart. Use them to work out what the parents would have given them, and use them to figure out what they would pass on to the next generation. So that's a clue for using pedigree charts. Right, so the next question. Is the disease associated with blue people caused by a dominant or recessive allele? That's the first part. And give a reason for your answer. And so if we go back to the diagram, we need to determine firstly if it's dominant or recessive, and then second, provide a reason. And so guys, firstly, we know that generally a dominant allele will be significantly predominant in the population. But that's not always true. So you cannot simply say by looking at the pedigree chart, because there are fewer individuals affected by this condition, that the condition is recessive. Because I can point to the example of genetic dwarfism. So we are recessive for that. In that sense, I mean people that are normal heighted, that are not genetic dwarfs, have recessive alleles. And if you look at the general population, you'll find that generally we all are normal heighted and we don't have the recessive allele. If we had the recessive allele, or if we had the dominant allele, beg your pardon, we would all be dwarfs. So that's an example of a dominant and a recessive allele which are not necessarily predominant in the population. So getting back to this, if we look at the distribution of the genotypes for Luna, now Luna is certainly affected by this individual, and if we look at Mabala and Maurice, both seem to be normal, okay, because they have an unshaded circle and square respectively. So the only way that Mabala can be a normal or not affected is if she has the dominant allele. And clearly from this diagram, as I mentioned, looking at Luna, we know that Luna has inherited the recessive allele from Mabala and also the other recessive allele from Maurice. And purely based on that, you know that in a heterozygous cross, when two dominant individuals are crossed that are heterozygous for a specific condition, you have a 25% chance of inheriting the recessive alleles. And that's clearly the possibility that has existed for Luna to have been affected by that. So clearly from this diagram, you can see that Mabala and Maurice are heterozygous, meaning that they are dominant for that condition, and that the only way that Luna can be affected by this condition is if she inherits it in a homozygous recessive combination. And hence we know that this condition is recessive, meaning that it's only expressed when the alleles are in a homozygous recessive combination. Okay, the next question is interesting, and I'm going to kind of see if we can get through this question through this section. Okay, I'm going to read through the question. It's obviously a genetic diagram. I'm going to give you some time to consider and ponder about this. When we get back, we're going to look at the solution to it. So I'll read through the question and then we can have a look at it. Mabala and Maurice both have normal skin coloring. Using a genetic cross, guys, show how this is possible for their son Luna to be blue. And this question is a 7-mark question. So we've got to show the possibilities between Luna and Mabala having this possible. So here is Mabala, here is Maurice, and we've got to show the cross between the two. So guys, I'm going to leave that for you guys to ponder over during a break, And when we come back, we possibly can look at your answers while I share my answers. Looney, over to you.
Thank you, Eugen. Guys, let's take a quick break and we'll see you shortly after this. Welcome back from the break, Matrix. I hope you guys are still enjoying the show. We are still assisting you to prepare for the upcoming exam. Thank you so much, Eugen, and over to you.
Thanks, Looney. So guys, I challenged you before we got into break to look at that question and see if you could put together a genetic diagram. I know that you probably started with it, but let's look at that question again, and I will help you to solve this. Remember that genetic diagrams most often come, and so it's a guaranteed question that you can kind of work on and understand the layout, because that's often marks that are given for something that's actually a learned concept. So you just lay out the diagram, you do a Punnett square, and you should be good to get the marks. Let's look at how we would solve this question. So again, I'm going to take you back to the question. The question relates to identifying the genotypes of Luna, the son, by doing a genetic cross, and look at the possibility of them having a son being blue. And so as I showed you that, we need to go back to the genetic diagram, and let's look at who we're looking at. So Mabala and Maurice are both what we call unaffected, meaning that they have got a dominant allele. But knowing that Luna is affected, she has to have two recessive alleles. This means that Luna is inherited a recessive allele from both of the parents. So it means that both the parents, Maurice and Mabala, are both what we say heterozygous for this condition. Now that's crucial to actually solving this genetic problem, is that we're able to identify the genotypes. And as I said at the start, use the carriers, use individuals that are affected by this condition or that have the condition to show or to determine what their parents have given to them or what they give to the next generation, which we clearly did in this example. So let's look at how I would have laid out a genetic cross. Now guys, there's different ways of laying out the possible gametes, and we're going to look at that. So I know that you guys were using cross diagrams at school. Some of us have looked at using Punnett squares. It really doesn't matter. The results are the same. It's the methodology of using a genetic diagram that's important. So let's look at one format of solving this question. So again, there are marks that are allocated for determining the phenotype. So as you see, the phenotypes of both the parents are normal skinned male or normal skinned female. In this case, we're not using their names. We're saying that the male was unaffected or having a normal skin pigmentation. And we identified the second thing as their genotypes being heterozygous, so we've got that. We then show the formation of gametes, and these gametes are indicated by splitting up the genotypes, and those are gametes. And we indicate that in circles. I often like to do that so that these are clearly different from a homozygous combination. We can show this possible offsprings by using a cross diagram, where we call the first crossed with the second, the first with the second, or the first with the first, and you would get these possible offspring. So you've got that as being one homozygous dominant to two heterozygous individuals to one homozygous recessive. So I refer to this as the F1 genotype ratio. We would then need to convert that F1 genotype ratio into the phenotype ratio. And in this case, the physical appearance of those individuals would be, we have these individuals, these three individuals having the normal unaffected skin color. And unfortunately, we've got that individual who has been affected with that condition. And we know that it is a condition in which homozygous recessive individuals are affected, and hence we know that this individual would have a skin color that is blue. So we've got to now interpret what is the possibility that we might have, or they would have had a child that has the condition. And if you look at the ratio, you've got 3 individuals that are affected to 3 individuals that are not affected to one individual that is affected, and that if you put it into a percentage, that is a 25% chance of having a child with blue skin, which essentially is significantly high. You have a 1 in 4 possibility every time that these couples would have had a child to have that child. So the marks are also allocated. If we look at the mark allocation, I'm just going to change the color of my pen so that we can see where the mark allocation for this is. So the mark allocation was for identifying the parents in terms of their phenotypes. The second mark is for the genotypes of the parents and correctly identified. Then you're going to be awarded marks for separating out the gametes and for showing the cross, and in the F1 genotype you're going to get a mark there, as well as putting together the phenotype ratio, and then most importantly is being able to illustrate a percentage or the ratio of those individuals that are born, in this case, with the affected skin condition. And there are marks for showing, as the examiners indicated, the P1 and the F1 generations or the crosses, as well as showing the process of meiosis and fertilization. So meiosis would be the division here that we're seeing in resulting in the formation of gametes, and fertilization would be the cross diagram. Right. I know that some of you choose to use the Punnett square, which I'm going to take the liberty of actually illustrating to you. And the Punnett square is essentially a very similar diagram, but it's more systematic, and the possibility of having made a mistake during the cross is significantly less. And I'll illustrate that and how simply it can be done. So again, the phenotypes are male, that's normal, male and female that are normal, but we know that their genotypes indicate that they are heterozygous, so that's the second mark that you would get. Again, we've got to show meiosis, and in this case, we've got to separate out these gametes, and then I often help myself to remember that these gametes are by circling them, and I've got to now transfer these into a Punnett square. The rule is that the pair need to go together, and this pair will then go into that row or column. It really doesn't matter if you swap in terms of if you swap the positions of where they go, but the important thing is that both these need to go together on one side of the table and the other two on the opposite end. And essentially this diagram is illustrating that this square is a result, let me get back to that this square is a result of a cross between that and that. So again, we write the dominant allele first, so that's capital N, capital N. This square is a result of a cross between that square and that, so it's the dominant and the recessive. And the last square is a cross between this, okay, and that, so that's going to be that. And then the corner square here, right at the bottom, is a cross between those two. And you would have identified that you have that individual, you've got one of those, and you've got two of the heterozygous individuals. So this is called a Punnett square, and this Punnett square is important in being able to identify, again, the genotypes of the F1 generation. So that then needs to be illustrated in an F1 phenotype ratio, and we also can show that in a genotype ratio, which would be, I'm going to illustrate that here just for now. So that's one of those to two of heterozygous individuals to one homozygous recessive individual. So we've got the F1 genotype ratio, and this is the F1 phenotype ratio, where we see that there are 3 normal, 1 child affected. Again, the same ratio. The difference is that we've now used a Punnett square to illustrate that, and we've used a cross diagram to show fertilization. You would be rewarded the marks for whichever diagram you choose, provided that you've done and applied the method consistently through the process. Let's move on to an interesting question, which I know that you guys have often kind of dreaded, and you did tend to do this, but let's look at an essay as a point of approaching the last part of the segment. Essays are there. They would always be in this exam, but remember that the essay is often broken up into subtopics. So I'm going to show you an example of an essay that was from a past exam paper, and this was an Eastern Cape paper prelim exam that was written in the year 2020. We're going to look at this topic. We're going to try and unpack the essay and try and break that down into small bits so that you can make this essay, which is kind of daunting when you sit and write this final exam, into smaller chunks that you can identify as easier to answer subsections. And that's essentially how we're going to do this, and you should be doing this for any other essay. So I'm going to read through the question and then unpack what the question requires that we do. So discuss the composition of the human karyotype. So there's the first part. We've got to discuss the composition of the human karyotype. Also explain why meiosis is important. Okay, so that's the second part. So we've got to now explain why meiosis is important. And thirdly, they talk about how non-disjunction during meiosis can affect humans. So if you look at this, there are three parts to this question. It's about the composition of a human karyotype. Second, we've got to talk about why meiosis is important. And finally, why non-disjunction occurs and the effects of that. So clearly, we can break down this huge essay topic into three little sections. And that's essentially being able to identify and then kind of allocate to yourself in your head, in your planning, as to what possibly be the mark allocations for those three sections. And looking at this, I know that an essay is always marked out of a mark of 17, where there are 3 marks for synthesis. And that synthesis mark is essentially for you to prevent it from being a flow diagram, but more specifically a cohesive essay where you've stipulated your points and there's flow and there's good synthesis and integration of your facts to explain a specific concept. And that's what we're going to show you today. Right, so let's get straight into a possible suggestion for this. Right, so when we look at the composition of the karyotype, a human karyotype has got 23 pairs or 46 chromosomes, so that's fundamental to that. And we know that karyotypes generally, if they're taken from a somatic cell, will have that many. We also know that if you're taking a karyotype from a gonad or from a gamete, my apologies, it would have half that, so it would have 23 chromosomes. So 23 in a gamete and 46 in a somatic cell. Okay? Karyotypes are arrangements of chromosomes where chromosomes are arranged in homologous pairs. So pairs meaning they have similar shape and size. We also can describe that a somatic cell has 22 pairs or autosomes, 44 of them, and we also have a pair of gonosomes or an X and Y chromosome for each cell. Autosomes are arranged in pairs from the smallest or from the largest to the smallest in a karyotype, with the smallest being your X and Y being arranged right at the end, and so we call these the gonosomes. So in a female, you'll have 2 X chromosomes. In a male, you'll have an X and a Y chromosome. So clearly, you would need to recognize the fact that males have an X and a Y chromosome, and females have 2 X chromosomes. We also need to discuss the comparison between the size of the X chromosome being significantly larger and the Y chromosome being significantly smaller, as illustrated here. So we have pairs of chromosomes, except the X and Y, which are not identical. They're quite different in size. So that is a possible 6-mark description for the significance of a karyotype and what its composition is. So the marks that you could get for several facts in this. The next part of the question was to discuss the importance of meiosis. Guys, we've discussed this. Meiosis is what drives change. So we know that evolution occurs because of variation. Where does that come from? That comes from meiosis. Fundamentally, the changes that occur during meiosis in terms of crossing over produces variation. And so meiosis is significant, firstly, in producing variation amongst the gametes, so that when there is fertilization between gametes, that the recombination creates variations from the parents in the offspring. So the significance of meiosis is that it produces gametes. And gametes, again, are haploid. So think of this. If gametes are haploid, when they fuse together, they restore the diploid number of chromosomes. So it's important that during gametogenesis that gametes are halved, and that when fertilization takes place, we return the diploid complement of chromosomes to the fertilized zygote. So meiosis is important in producing variation. Second, in reducing the chromosome number, so that when fertilization takes place, we maintain the diploid composition of chromosome numbers. And thirdly, if you look at the significance of meiosis, it allows for genetic recombination during a process called crossing over. Yes, in prophase 1, we know that crossing over occurs during meiosis. And that crossing over creates variation in the characteristics that are inherited from parents to offspring. And so we know that there's a constant variation that occurs because of crossing over. So that's the second part of the question, and we'll move on to the third part. And that third part tells us about the non-disjunction in humans. Okay, and so if we look at non-disjunction, essentially we're discussing how aneuploidy results in conditions such as Down syndrome, where there is non-separation of chromosomes. So guys, anaphase 1 is when chromosomes move apart. In anaphase 2, we know that the chromatids pull apart. If that process does not occur, you'll find that there's an uneven breaking of chromosomes, resulting in a full chromosome having 2 chromatids being pulled to one side. And if that cell fertilizes with a sperm or egg, it will result in that resulting cell having an extra chromosome or having one less chromosome. And that abnormality is called aneuploidy. And examples of that would be Down's trisomy, or conditions such as Klinefelter or even Turner syndrome, which are results of aneuploidy. Guys, I've got to get to the challenge question very quickly, and I think we're almost out of time, but I'm going to very quickly give you the response. So most of you seem to have responded with the option of B, and I'm going to go back to that question and look at B. So B was the preferred answer, and this seems to be correct. Let's go to that question and see what it was. So here you had to identify, let me go back to that, the homozygous antennae as well as the shape for the heterozygous body parts. And if you applied your understanding, guys, to that, so homozygous antennae would be having similar, so normal homozygous antennae, and then it had a heterozygous combination for body stripes without stripes, so this would be the heterozygous combination for body stripes. So the most obvious answer here was homozygous for normal antennae and heterozygous for body stripes. And clearly these were the possible combinations that were present in fly 2. So the answer here for this was fly 2, and if you did get it, well done. It is obviously a question that requires lots of processing, and you need to apply that understanding. So guys, that's a wrap for my side. We're almost done. Wish you all the best for your exams. I'm going to hand you over to Looney.
Thanks, Eugen. Matrix, thank you so much. It's been a great show. We're wishing you everything of the best for the upcoming exam. Remember, we do have study resources on our platform, so please check those out. A big congratulations to all of our competition winners who will be announced after the show. Don't forget to check out our schedule on www.wasamatrix.co.za. From me, Looney, Nicolene and Eugen, thank you and goodbye.
2020. Congratulations for being so brave, so committed, and for staying the course during one of the most difficult years in our history. We're here to support you all the way. Our Was a Matric campaign was set up to support you up until the 8th of December 2020. We are offering you interactive exam guides, shows on SABC3, DSTV, Catch Up, and OVHD channel 122 that are not to be missed. I wish you all the best in your final exams. You are the hope and the future of our country, and we're so proud of you for coming this far. No matter the outcome, you are at the finish line. Now give it your very, very best. And I thank you.